Hmm. Hello students, welcome to Trunkie Academy. We are studying about the prokaryotic cell and eukaryotic cells. Okay, cell as the structural and functional unit of life. In this regard, we studied about yesterday about the prokaryotic cells, the different parts of the prokaryotic cells, and we also studied about the bacteria. Okay, because bacteria is the important one of the important example. You can say the major living organisms which is made up of prokaryotic cell. Okay, bacteria is the bacterial cell is prokaryotic cells, and with regard to that we studied about the different parts of the bacteria, the bacterial cell wall. Okay, and the types of bacteria based on the strain they take, based on the shape. Also the bacteria are classified into four groups. Okay, that is about the bacilli, cocci, coccus or cocci, and then about the spirillum or vibrio. Okay, all this that we study. Now today we'll study about the eukaryotic cells. As I told you several times and repeating once again that a prokaryotic means the primitive type of cells. Means the pro means primitive and the karyon means the nucleus, means We are talking about the nucleus, which is a primitive type in prokaryotic cells. Where in the prokaryotic cell there is no distinct prominent nucleus, and the chromosomes are dispersed in the cytoplasm. So genetic materials are naked and chromosomes are dispersed in the cytoplasm. Okay, but in case of eukaryotics, eukaryotic means EU means the true, and karyon means the nucleus. So U is equal to true, and the karyotic word has been come from the karyon, and karyon in the nucleus. So in case of the eukaryotic cell, the nucleus is true. There is a membrane-bound nucleus is there, and the chromosomes are lodged well within the nucleus. Okay, and then even the chromosomes are also little advanced, and they have got histone proteins that we'll study in another chapter. Now eukaryotic cells means that includes all the protists, plants, animals, and fungi. So when we talk about the prokaryotic, when we study about the prokaryotic, we observe that bacteria, mycoplasma, cyanobacteria, and the PPLO, they represent the prokaryotic cells. Okay, but there's a very small group of organisms. But they now in eukaryotic, all the plants, okay, all the green plants, all the fungi, all the animals, including protozoans, and the protists means that means protozoans and the single cell, all the single cell, but they are eukaryotic cells like amoeba, paramecium, plasmodium, etc. Okay, so the eukaryotic cells include all the protists, plants, animals, and fungi. Okay, in eukaryotic cells there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Unlike prokaryotic cells, in case of the eukaryotic cells, there is compartmentalization of the cytoplasm, and the membrane or cell organelles are membrane bound. Okay, and the well defined cell organelles are found, and the membrane bound cell organelles are found in case of the eukaryotic cells. Okay, the compartmentalization of cytoplasm we find in case of eukaryotic. Okay, this is the advanced type of cells. Okay, eukaryotic. Now in case of protists, this is the example. Okay, unicellular eukaryotic organism. Protists mean unit. In case of prokaryotic, they are also unicellular, but they are prokaryotic. Okay, here protista is the monera. Sorry, monera is the unicellular and but prokaryotic. Kingdom monera. Okay, the 5 kingdoms are there classified by the R.H. Whittaker. Kingdom monera, kingdom protista, kingdom fungi, kingdom plantae, and kingdom animalia. Okay, so kingdom monera means prokaryotic cells, organism with prokaryotic cells. That includes bacteria, cyanobacteria, mycoplasma, and PPLO that we studied yesterday. In case of eukaryotic cells, these protista or kingdom protista are also unicellular like monera, but these are of eukaryotic cells. Unicellular but eukaryotic. Monera unicellular but prokaryotic. Protista unicellular but eukaryotic. Like euglena, paramecium, plasmodium, amoeba, etc. Okay, that's one. Now this is the euglena verdis. Euglena, it's got flagella. Okay, this is the paramecium. Okay. Now plants, all the plants. Okay, whether it is dicotyledonous, whether it is monocotyledonous, whether it is gymnosperm, whether it is angiosperm, whether it is pteridophytes, whether it is the bryophytes, or whether it is the thallophytes, algae, whether it is the trees, whether it is shrub, whether it is the herbs, small plants, all the plants, they are made of the eukaryotic cells. Okay, so then the animals, okay, whether it is the class Mammalia, whether it is the reptiles, whether it is the what we call the amphibians, or the fishes, okay, or the other animals like the starfish, arthropodons, okay, or the mollusk snail, or the small worm-like animals, or any other type of animals, that all are made up of the eukaryotic cell. Okay, fungi, kingdom fungi, also the fungus, they are also made of the eukaryotic cells. They are not photosynthesizing plant, okay, but they are also made of eukaryotic cells. Okay, so this is placed under a separate kingdom. Okay, so kingdom fungi. They are also made up of the eukaryotic cell. What makes a cell eukaryotic? An organized nucleus with a nuclear envelope or nuclear membrane. Okay, an organized nucleus. There is a chromosomes, and chromosomes are also well organized. Okay, with nucleosome and histone protein. Okay, so in class 10 you have little bit studied about the histone and the nucleosome. Okay, and this presence of nucleosome and the histone protein makes it well organized, and the nuclear envelope is there, and this makes the eukaryotic cells eukaryotic. Okay, eukaryotic cells possess an organized nucleus with a nuclear envelope. Eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. These cells have the eukaryotic cells have a variety of complex locomotory cytoskeletal structure. That means it may be the cilia, it may be the flagella, or it may be some other complex structure like a pseudopodia, as we observe in case of amoeba. Okay, in amoeboid movement. Eukaryotic cells is genetic material. In eukaryotic cell, the genetic material is organized into chromosomes. So that is not naked and organized into well within the chromosomes. Okay, all eukaryotic cells are not identical. Okay, that means there are so many different variety of eukaryotic cells are found. There are great variation in eukaryotic cells of plants and animals. Plants and animals are different. Okay, and we see the distinct difference between the eukaryotic cells of the plant and eukaryotic cells of the animals. The most prominent feature and the difference is the presence of cell wall in plant cell and the absence of cell wall in plant cell. The vacuoles are bigger in size and fewer in number in case of plant cell. Vacuoles are smaller in size and little more in number than the plant cell in case of the animal cells. It's found like this. There is a centriole and centrosomes in the animal cells, which is not found in the plant cells. Okay, so there are some differences between the plant and animal cells, although both are the eukaryotic cells. Okay, so plant cell possesses cell wall, plastids, and plastids is found in plant cells only because this is a pigment. The three kinds of plastids are found. That is leucoplast, chromoplast, and chloroplast. Chloroplast is the green pigment containing cell organelle which has some chlorophyll pigment for photosynthesis. So large central vacuole is also present. So these are the important distinguishing feature of the plant cell, which is not found in animal cell. Okay, so centrioles are absent in almost all plant cells, and cell walls, plastics, and large central vacuole are absent in animal cells. So this is one of the interesting feature and the character that we find in plant cell and animal cell, the difference between these two types of the cells, that is the cells of the plant cell and cells of the animal cells. Okay, centrioles are present in almost all animal cells that helps in the cell division. Okay, centrioles are the cell organelles. The two cell centrioles are arranged perpendicular like in T cell, and they form some structure, and they help in the movement of the chromosomes during the cell division. Okay, so cell membrane. Now we come to the point. Now we see there are different types of cell organelle in the eukaryotic cell. Okay, and in eukaryotic cell, when you observe the eukaryotic cell, we know that first there are so many different organelles are there. But when we say study about the different parts of the eukaryotic cells, we see that there are well-defined cell organelles are present and eukaryotic, like Golgi complex or Golgi bodies, endoplasmic reticulum. Okay, ribosomes. Ribosomes are also present in prokaryotic cell that you studied yesterday, and there are some important cell organ like mitochondria is also present in eukaryotic cell. But first of all, we need to study about the plasma membrane or cell membrane. That is the limiting membrane, that is the within which cytoplasm are present or protoplasm is present, and cell membrane is the boundary. Okay, limiting. Okay, so the detailed structure of membrane was studied only after the advent of electron microscope, and the microscope invented by the Knoll and Ruska, 1950. So just imagine. Okay, microscope was for the first time invented by Antony von Leeuwenhoek, but that was very simple and not much magnifying. Okay, the magnification power was not much as the electron microscope is a very advanced type of microscope where we can magnify the object 100,000 times bigger. Okay, but this was discovered in 1950. So detailed structure of the plasma membrane or cell membrane was studied only after the discovery of electron microscope, and these electron microscopes were discovered by Moll and Ruska. These are the name of the scientists who discovered the electron microscope in 1950s. Okay, in the decade of the 19. Now, before this discovery of the electron microscope, there was some idea, there was some concept in the mind of the scientist. Okay, what is a cell membrane or plasma membrane, and how it is made up of. Okay, so the chemical studies on the cell membrane, especially in the human RBCs, enabled the scientists to deduce the possible structure of plasma membrane. So study the detailed structure of plasma membrane or cell membrane. Okay, the scientists, okay, the biologists, they use the RBCs, red blood cells, erythrocytes of the human, and they observed the plasma membrane of the RBCs, and they were able to understand and deduce the possible structure of the plasma membrane, the composition of the plasma membrane, the detailed structure of the plasma membrane by studying the plasma membrane of the red blood cells or erythrocytes. Okay, plasma membrane is a bilayer structure made up of the lipoprotein, lipid, and protein. There are some channels and porins. Okay, and there are some kind of carbohydrate chains attached on the this protein of surface, where this plays an important role in its particular kind of identity. Okay, that we'll study later on. The cell membrane is composed of lipids that are arranged in a bilayer. Okay, so two layers are there of lipids are there. Okay, like here you see the lipids are arranged within the membrane with a polar hydrophilic. Okay, so hydrophilic means the outside one, the outside which is facing the outside environment, and also the one which is facing the cytoplasm. So these are the hydrophilic and head. So that region which is hydrophilic is the head, hydrophilic head, which are outside, and hydrophobic tails. So there are two parts basically. So in case of the bilayer, and we find this that there are the head, okay, and the hydrophilic head and the hydrophobic tail. Okay, so they are facing towards each other, the bilayers. Okay, so they are, because they are hydrophobic, so they are arranged facing each other, and hydrophilic heads are facing the outside environment and the cytoplasm. So they are arranged in this, and you can see one kind of a substance that is moving. Okay, that is the protein, and on that protein there may be this is called phospholipid bilayer. Why phospholipid? The lipid is the phospholipid, a type of lipid. Okay, phospholipid. So this is the phospholipid, and this the layer is made up of the phospholipid, a type of lipid, and hence it is called the phospholipid bilayer, since these two layers are present. Okay, so you knew that there are hydrophobic head and hydrophilic head and hydrophobic tail. Okay, so this is the structure of the so hydrophilic head and hydrophobic tails are present. So hydrophobic tails are towards the inner part facing each other, and hydrophobic, the head region, one is facing towards the external milieu, and one is facing the cytoplasm. Okay, now the cell membrane in detail we'll study about. Okay, because this is the structure. It's not very complex. It's not a very simple. It's a layer of a kind of chemical substances. Okay, it is a very complex and very important part of the cell, because it plays a very important role in the movement of the molecules in and out of the cell. Okay, entry and exit of the cell. There are some substances which are not needed by the cell, and there are some substances which the cell needs. So depending on the need of the cells, this cellular membrane or the cell membrane or plasma membrane acts selectively. Okay, it selectively permits some substances to enter into the cell, and some substances not to enter, depending on the requirement of the cell. Okay, so this ensures that the non-polar tail, I mean the hydrophobic and hydrophilic part of the the bilayer, phosphor bilayer, ensure that the non-polar tail of saturated hydrocarbons are protected from the aqueous environment because they are facing each other. So they are in no longer they are not in contact with the aqueous environment because they are hydrophobic, whereas the hydrophilic heads are in contact with the aqueous environment. They are facing their cytoplasm as well as the external environment, so they may face the aqueous environment. So since they are hydrophilic, they may face, but to ensure the hydrophilic tails are not in contact with the aqueous environment because they are hydrophobic, so they are facing each other. Okay, so they are made of the saturated hydrocarbons. The tails are made up of the and these saturated hydrocarbons, okay, are hydrophobic in nature. Okay, chemically, lipid component of the membrane mainly consists of phosphoglycerides. Like I told you, why this is called phospholipid bilayer, because the lipid that is present in the membrane, the type of lipid, that is the phospholipid, a kind of lipid that we'll study in biochemistry, and the part of when we study about the biomolecules. When you study the chapter biomolecules, that time you will study about the this lipid, carbohydrate, protein, etc. Okay, so chemically, lipid component in the membrane consists of the phosphoglycerides. Okay, and the cell membrane also possesses protein and carbohydrate. So we find all the three different types: protein, carbohydrate, and lipids. Okay, the membrane of RBC has 52% of protein and 40% of lipid. So this is the composition, okay, percentage wise between the protein and the lipids. Okay, so proteins are a little higher in composition, you can say in concentration. So 52% are the protein and 40% are the lipids. Okay, the membrane proteins are integral or peripheral. Integral means they are well embedded, they are passing across the membrane, the two bilayer, and the peripheral, they are just above. Okay, not fully embedded. Okay, the peripheral proteins lie on the surface of the membrane, while integral proteins partially or totally buried in the membrane. So when the peripheral, that is just above, just on the surface, okay, and integral means they are passing through the membrane or they are partially embedded or buried in the membrane, like it has been shown in this picture. You can see the integral protein, which is transmembrane protein. Just this one, okay, integral protein, this one. Okay, this is passing through the, okay, across the membrane. Okay, so this is the called the integral protein, whereas this one you can say is just embedded partially, you know, in the, so that will be this is also the integral, but here in case of this one, this is the on the peripheral surface, so that is the we call that as the peripheral protein. Okay, but this will also be considered as the integral, but since it's partly embedded. This will be also integral. Okay, this one also integral. An improved model of the structure of the cell membrane was proposed by Singer and Nicholson, yeah, in 1972. This is one of the in 1950, the Knoll and Ruska discovered the microscope, and after that study began. Okay, after the discovery of the electron microscope, the study of the different cellular structure, okay, began. And Singer and Nicholson were a very famous scientist. They studied extensively, okay, and they studied about the structure of the plasma membrane, and they after very intensive study, they came to a model, okay, and they named this model as the fluid mosaic model, okay, and this widely accepted. Before this, there were some other models and some other type of structure were predicted, but that was rejected by later on the other scientists. So Singer and Nicholson. So these are the two scientists who actually worked on the structure of the plasma membrane or cell membrane, and then they came to a structure as of the plasma membrane as fluid mosaic model. Okay, so what is this fluid mosaic model? We'll see this also. Fluid mosaic model means when this is a kind of fluid, where is a mosaic. Okay, there are some kind of embedded protein and they are moving. Okay, so that will study carbohydrate protein chains. You can see on the surface of the protein. Okay, you can see the non-polar region. Okay, and the globular protein is there, that is passing through the plasma membrane, and the cholesterol is there, made of the hydro phobic part, other tail, and the phospholipid, you can see phospholipid head. Okay, so these are the different chemical constituents. The lipids are quasi-fluid, yeah, not semi-fluid but quasi-fluid. Okay, quasi means one fourth. Okay, quasi-fluid enables lateral movement of proteins with bilayer. So these lipid, this cell membrane are lipids quasi-fluid in nature. Okay, so lipids that is present in the plasma membrane is the quasi-fluid, enables lateral, so that the movement of proteins in the bilayer. Okay, so that is lipid fluidity explains the movement with membrane. Okay, so they explain the movement with membrane because there are since there is some kind of fluidity in the membrane, so there is change movement, a movement of the cell is also possible. Okay, the fluid nature of the membrane is also important for cellular function. Yes, the fluidity and the fluid nature of the membrane is very important with regard to different cellular functions. Okay, so when you say cellular function, it says about the cell growth, secretion, endocytosis, and cell divisions. Okay, so these are and formation of cellular junctions. So these are the important aspect, or you can say functions, due to fluidity of the plasma membrane. Okay, or flexibility. If it is very hard, rigid, and then not movement, so this will be not cell growth. Okay, since it is expanding little bit, the cell growth is possible. Secretion, endocytosis. Okay, means some kind of the molecules or some kind of substances are coming to the plasma membrane that is taken in endocytosis by the cell division, dividing of the cells. All these are possible due to fluid nature of the membrane, and formation of cellular junctions, or the junctions between the two cells. Okay, so the transport of molecules without requirement of energy is the active transport. Sorry, without energy it is the passive transport. But the energy is required, that is the active transport. The transport of molecules without requirement of energy. Okay, that means the molecules is transferred from higher concentration to the lower concentration. Okay, so that is the for example, if there is a molecules present here, okay, and there is a membrane, okay, and the molecules are moving, okay, here, passing through, because the lower concentration of molecules are present of this type of molecule, so they will come from the higher concentration to lower concentration. This is called the passive transport, passive movement without requirement of energy, and when the energy is required, it's called active transport. Okay, so this is the passive transport. Now, one of the most important functions of the plasma membrane is the transport of molecules into and out of the cells. Okay, plasma membrane acts as the gate and also for the movement of the molecules from in and out of the cell. So membrane is selectively permeable to some molecules present on either side of it. Okay, so it is permeable to some molecules, and so it is selectively permits, okay, the substances to enter into or go out of the cell. Okay, so many molecules can move freely across the membrane without any requirement of energy. This is called the passive transport. Some molecules are there that can move in and out of the cell freely without any requirement of energy. That is the passive transport. Okay, neutral solids may move across the membrane by the process of simple diffusion along the concentration gradient. So when I told you this is a concentration gradient, when I drew this diagram, the molecules are present, the concentration. So when the molecules are moving along the concentration gradient from higher concentration to the lower concentration, this is called concentration gradient. When the molecules are moving along the concentration gradient, then from higher to lower, then it is a passive transport or passive movement. Okay, so this takes place in the neutral solids. Okay, not the ionized. Okay, so here you can see one diagram, one picture where the molecules are going into, and then it's a kind of a channel or chamber where it's entering and then it's opening when it moves to the outside. Okay, or when it comes in from outside and then moves into the outside. Okay, so diffusion takes place from higher concentration to the lower concentration, and facilitated diffusion means that some kind of molecules, some substances, they pass through a particular channel which is present across the membrane. Okay, so they are passing through a particular kind of channel or protein channel. Okay, or some kind of channel that is present through which they are entering into the cell. Okay, along the concentration gradient from higher concentration to lower concentration. That is called the facilitated diffusion, because this is facilitated by a particular type of channel through which these molecules are able to enter from higher concentration to lower concentration. This is both are both are the both diffusion and this facilitated diffusion are passive transport. Okay, passive transport. And from higher the transport takes come from the higher concentration to lower concentration, and this is called the passive transport. What is osmosis? Movement of water by diffusion across the membrane is called osmosis. Now this is very interesting. The process by which plants are able to absorb water from the soil, that is through the root hairs, that is the osmosis. This is a very important. Okay, so in case of the osmosis, movement of water by diffusion across the membrane occurs. Okay, so when the water molecules are diffusing in to the root hair cells by the process of this is the osmosis. Okay, the polar molecules required a carrier protein of the for the membrane to facilitate their transport across the membrane. Polar molecules, not neutral molecules, but polar molecules, they require some kind of carrier. Like I told you, there is a kind of protein channel like here. So we require a carrier protein. In this case, you can see this is a carrier protein. Okay, so they require a carrier protein and of the membrane to facilitate the transport across the membrane. Some ions are transported across the membrane through its carrier protein. Okay, because it cannot simply pass through, and they require against the concentration gradient, but from lower to higher. This requires requirement of energy. Sometimes some ions need to be transported against the concentration gradient from lower concentration to higher concentration, and that requires the involvement of the energy. Okay, such a transport is an energy dependent process in which ATP is utilized and is called active transport. Unlike passive transport, here there is the requirement of energy, and this is against the concentration gradient, that is from lower concentration to higher concentration. Like I have already explained in one class, and I'm explaining that when there is a kind of in an inclined plane where rolling down some things, it will automatically roll down because it will go towards the gravity according to the gravitational force. But when you have to move up, roll up, okay, we are against the gravitation, that time it requires energy, right? Similarly here also, this analogy helps you to understand that active transport, in active transport, where there is the transport of molecules from lower concentration to higher concentration, that it requires the involvement, or it is a energy dependent process where ATP is utilized for the transport of molecule in active transport. Okay, so here you can see the ATP is there, now it is moving. The concentration of that yellow is stuck in the inside of the cell, the extracellular fluid. Okay, so this yellow spherical substance that is depicted as the sodium ion. So if you observe that the sodium ion are present lesser in the extracellular fluid, and the sodium ion, the concentration of the sodium ions are more in the inside of the cell. So what will happen? So this has to move against the concentration gradient from the higher concentration, from the lower concentration to higher concentration. You can see, okay, that's going upward. So what will happen? From the ATP is there, and then with the help of this, it is going to the passing through the plasma membrane with the help of a protein channel and the involvement of the ATP. Okay, so from lower concentration, so the concentration of the sodium ion are less inside the cell, and outside the cell it is more, but from lower concentration inside the cell, they are moving to the outside of the cell because of the active transport. Okay, now diffusion is going from higher concentration to lower concentration, the passive transport, and in case of the active transport is the requirement of energy, ATP. So they are moving from lower concentration to a higher concentration. So this kind of movement or transport of molecules are found across the plasma membrane, that is a diffusion, facilitated diffusion, and active transport. Both diffusion and facilitated diffusion are the passive transport because that do not require expenditure of energy. Okay, only the difference is between the diffusion and the facilitated diffusion is that in case of facilitated diffusion, there is a kind of channel or the protein channel through which the molecules are moving in or out. Okay, along the concentration gradient. But in case of the active transport, it is the energy that helps to drive the molecules from lower concentration to higher concentration. Okay, now I'll go through some important questions. Okay, quickly. Every living cell has the food vacuoles or chloroplast or cell membrane or cell wall. All living, just imagine this. Chloroplast is present only in plant cells, so that is not correct. Food vacuoles are not present in all cells, so that's not correct. Okay, cell wall is present only in plant cells, so it's not present in all cells. So correct answer for this question will be option number 3, that is the cell membrane. Okay, so cell membrane is present in all cells. So the size of a typical eukaryotic cell is about 20 micron to 30 micrometer, or it is a 10 micron to 20 micrometer, or 5 micron to 10 micrometer, or 10 micron to 15 micrometer. So it is about a 10 micron to 20 micron. This is the size of the typical eukaryotic cell. Okay, there may be variation, there are variations. Okay, but this is about the typical eukaryotic cell. Okay, prokaryotic cells are much smaller than this, but eukaryotic cell size is about 10 micrometer to 20 micrometer. Now, widely accepted model of plasma membrane is the fluid mosaic model, trilamellar model, the previous segment. This was not. So then, when the scientists studied about the trilamellar model given by some scientists, okay, previously, they found some kind of drawbacks in the model, and they were not satisfied with this kind of. Clover leaf model was also given, but this was also not satisfactory. Sandwich model was also, to some extent, yeah, it was understandable, but there was also some drawback. And then the fluid mosaic model is widely accepted, given by the Singer and Nicholson, the scientists in 1972, because these two scientists played a very important role for understanding the structure of plasma membrane. And this understanding the structure of plasma membrane led to so many important discoveries and invention, and also the different kinds of biotechnological improvement. Okay, and also some other discoveries in the medicine, in agriculture, and other field of the science. Okay, because by knowing this, there are so many other things to discover, discovered depending on the structure of the fluid mosaic model. Okay, so fluid mosaic model of plasma membrane was proposed by Robertson, Davison, Singer and Nicholson, and Singer and Nicholson is the correct answer. Okay, so this is important. Now, according to the fluid mosaic model, the cell membrane is a solid layer, or liquid layer, a fluid layer with solid particles, or semi-solid layer. Just try to understand. It is the solid layer, or fluid with solid particles, or it's liquid layer. It is the fluid with solid particles. Okay, because it is fluid like quasi-fluid, but the proteins of fluid particles are moving. Okay, they are floating. Okay, so this is about the third option is the correct answer. Okay, so now this was all about the structure of plasma membrane, and we started with the eukaryotic cell. Eukaryotic cells are the advanced type of cells which have the true nucleus, that is the prominent and distinct nucleus, and the nucleus has got nuclear envelope, nuclear membrane, and the chromosomes are also well organized within the nucleus, and there is a nucleosome and histone protein. Please remember this will be studied in another topic, where a nucleosome. When you study about this, then I will describe in detail. And histone protein made up of the octamer of histone protein. Okay, this is not found in prokaryotic cells. So these are present, and after this we studied about the plasma membrane. We have to study about the other cell organelles also, but first of all we need to know about the plasma membrane. There is mitochondria, there is Golgi bodies or Golgi complex, there is endoplasmic reticulum, ribosomes, okay, and centrosome. But first of all, the membrane that is enveloping the cell, a cytoplasm or protoplasm, that we need to study, and that we study that this plasma membrane is made up of two phospholipid layer, that's called phospholipid bilayer, and some proteins are embedded, and on the surface of some carbohydrate chains are present. Okay, so this is how, and this is the lipid is the passive fluid, and the proteins are moving. Okay, they're floating, and there is the transport of substances. This plasma membrane is selectively or semi-permeable, ensuring that only those substances will be move in and out which is suitable for cell. Okay, so this was about the plasma membrane. And so in the next class we'll study about the next topic. Okay, and tomorrow is Sunday, so holiday, and day after tomorrow we will meet again at the same time. Okay, as I told you earlier that we are sending you the DPP for your practice to today also we'll send the DPP when you log on to your academic booster, and you will click on the resources, and then you click on the writing material. Okay, you'll find DPP on the topic that we have discussed today. Okay, so you download that DPP, click on the DPP and download, and go through the question and answer those questions and send back to us for our evaluations. Okay, so thank you for joining today and listening very patiently, and then we will meet again day after tomorrow at the same time. Okay, so till then, goodbye, take care. Okay.